We're mostly focused on trying to assess the T-cell receptor peptide MHC interactions. So we design MHC multimers, meaning that it's MHC class 1 complexes loaded with peptides of different origin of interest. And we have some high throughput techniques to generate these in large numbers, which means that we can cover or generate these reagents based on patient HLA types, at least to a large extent. So what we do is that we, for example, for a given patient cohort, identify peptides of potential interest in the certain disease settings, and then we generate reagents matching these based on the HLA expression of the patient. So in a bit more details, what we do is that we have a UV sensible peptide that is cleavable, so we can basically exchange peptides sitting in the groove from a standard complex carrying this conditional ligand to any peptide of interest. So by this means we can generate large libraries of peptide MHC reagents in a relatively short period of time, and then we can create these reagents for which we can actually stain T cells from tissue samples. We have techniques for which we firstly label these reagents so that we are able to mix many in single samples, and we are also further purchasing this route to get even higher complexity in our assays. But currently, for example, we have recently, in collaboration with people in the UK at UCL, Charles Swanton and Sari Kusara's group, worked in non-small cell lung cancer study where we looked at whether T cells would recognize changes occurring in tumors upon mutation changes, and specifically focused on mutation that arises in what you can say it in the trunk of the tumor, so that it's like clonally distributed in all the tumor cells. So you can basically really hit the core of the tumor, and we can see that we have specific immune reactivity specifically directed against those type of mutations. We are definitely working to increase the complexity of these techniques, and what we believe that we can do then is that we can much better go into different types of patient material, for example, to find these new mutation-derived new epitopes also in peripheral blood and in tissue biopsies without having to expand T cell at first, for which you always introduce some bias to your T cell repertoire, so we can better mimic basically what is really happening in the tissue with these more advanced techniques that we're working on. And so far what we do is more like in an exploratory phase where we haven't screened like large patient cohort, but for detection of these mutation-derived new epitopes in cancer, we specifically see that within this study with in lung cancer, together with Charles Swanton, that we see this reactivity primarily directed against these clonal antigens, and that so far is pretty significant, and we haven't seen any responses that are directed against these subclonal mutations. So there seem to be some characteristics, but we don't know why this is, whether it's a matter of expression level or it can be other reasons why these mutations are predominantly recognized by the immune system. I think at least in this direction of understanding immune recognition of cancer, I think what we really need is to have better predictive tools so that we can really find out in this huge landscapes, for example, of mutational changes, which are the ones that are likely to be immunogenic, because now we are really looking for the needle in the haystack, whereas if we would have better ways to focus our libraries, we would have a much better chance to find them, but also a better way to have predictive tools.